Hey everyone, this is Andrew. In this video, I'll be discussing the cell cycle, mitosis, which is nuclear division, and cytokinesis, which is when the cell splits in two. The cell cycle, mitosis, and cytokinesis. This is the cornerstone of biology. The cell cycle and mitosis forms the framework for biological study. It's going to be the base from which you learn every other topic. A cell reproduces by carrying out an orderly sequence of events in which it duplicates its contents and then divides in two. This cycle of duplication and division is known as the cell cycle. It is the essential mechanism by which all living things reproduce. So in this mechanism of how cells reproduce, we have to consider three major questions. One, how do cells duplicate their contents, including the chromosomes? And two, how do they partition the duplicated contents and then split in two? And lastly, how do they coordinate all the steps and machinery that is required to facilitate this division? This third question I will save for another video where I'll talk about the regulation of the cell cycle. So the eukaryotic cell cycle usually includes four phases. Seen in a microscope, the two most dramatic events in the cell cycle are when the nucleus divides, which is called mitosis, and when the cell itself splits into two, that is called cytokinesis. These two processes together constitute the M phase of the cell cycle. In a typical mammalian cell, the whole of the M phase usually lasts about an hour. The period between one M phase and the next is called interphase. During S phase, or synthesis, the cell replicates its DNA, and S phase is flanked by two gap phases, the G1 phase and G2 phase, during which the cell continues to grow. During these gap phases, that's when the cell monitors both its internal and external environment, ensuring that the cell has suitable conditions for reproduction and that preparations are made so the cell can move forward with division. During the entirety of interphase, just remember that the cell continues to transcribe genes, synthesize proteins, and grow in mass. So as I stated before, I'll discuss it in a whole nother video, but to ensure that the cell replicates all its DNA and organelles and divide in an orderly manner, eukaryotic cells possess this very unique cell cycle control system, which guarantees that the events of the cell cycle, including DNA replication, mitosis, and so on, occur in a very set sequence and that each process has been completed before the next one begins. Cell proliferation in animals requires both sufficient nutrients and specific signal molecules in the extracellular environment. If these extracellular conditions are unfavorable, cells will delay the progress through the G1 phase, will most likely enter a resting state, which is known as the G0 phase. That's why I've drawn this with like a little dial, a little controller, because the cell cycle control system acts like a control. So now let's move into each step of the cell cycle and cell division, starting with interphase. So during interphase, the cell will increase in size, the DNA of the chromosomes will be replicated, and the centrosome is duplicated. So let's look at this cell. So what we see here, we have the microtubules which are growing from the duplicated centrosome. That's what this is right here. Notice that the chromosomes in the nucleus are decondensed, which is extremely important as we move forward. The nuclear envelope, we see that the nuclear envelope, its pores are open. We got the open pores right here. But realize that the nuclear envelope is still intact. It's still there. So as we move into prophase, at prophase, the duplicated chromosomes, each consisting of two closely associated sister chromatids, will condense. Outside the nucleus, the mitotic spindle assembles between the two centrosomes, which have begun to move apart. For simplicity, only 3 chromosomes are drawn. But we see here that there's these things called kinetochores, which attach at the middle of the condensed chromosomes, and the mitotic spindle is forming at the same time that these chromosomes are condensing. So we move to the next one. Prometaphase comes after prophase. Prometaphase starts very abruptly with a breakdown of the nuclear envelope. Chromosomes can now attach to spindle microtubules via their kinetochores and undergo active movement towards the poles of the cell. We see here that the fragments of the nuclear envelope are away from the chromosomes and that the kinetochores are connecting and the chromosomes begin to start moving. And that's where metaphase, the third step, comes in. At metaphase, the chromosomes are aligned at the equator of the spindle, midway between the spindle poles. The kinetochore microtubules on each sister chromatid attach the opposite poles of the spindle. So if it helps you to remember, I remember metaphase corresponds with M, so they meet at the middle, the chromosomes meet at the middle or at the metaphase plate. So the next step is anaphase. At anaphase, the sister chromatids will synchronously separate and are pulled slowly toward the spindle pole to which they are attached. The kinetochore microtubules get shorter and the spindle poles also move apart, both contributing to chromosome segregation. Following anaphase, we have telophase. During telophase, the two sets of chromosomes arrive at the poles of the spindle. A new nuclear envelope will reassemble around each set, completing the formation of the two nuclei, ending mitosis. So the division of the cytoplasm begins with the assembly of the contractile ring, which forms right at the middle, at that cleavage furrow in between the two cells. It's the same, it's one cell still. However, the cleavage furrow will form, and that's where the two cells will then separate. This separation occurs during cytokinesis. During cytokinesis of an animal cell, the cytoplasm is divided in two by a contractile ring of actin and myosin filaments. This pinches the cell into two daughter cells, each with one nucleus, and there we have a completed cell cycle, and these two cells will then go on to do the exact same thing when the time is required. So in order to understand the mechanism of the separation of the replicated chromosomes, the chromatids, via the kinetochore microtubules, let's first remember that prometaphase starts extremely abruptly with the disassembly of the nuclear envelope. This breaks up the envelope into small membrane vesicles. This is actually triggered by the phosphorylation and consequent disassembly of the nuclear pore proteins and the intermediate filament proteins of the nuclear lamina. Because of that, the spindle microtubules can gain access to the duplicated chromosomes. The spindle microtubules will then attach to the chromosomes at their kinetochores, these protein complexes that assemble on the centromere of each condensed chromosome during late prophase. So once the nuclear envelope has been broken down, a randomly probing microtubule encounters a kinetochore, right? It'll bind to it, capturing that chromosome, linking it to the spindle pole, and because these sister kinetochores face in opposite directions, they tend to attach the microtubules from opposite poles. That's why we get the separation towards each pole. It's called bi-orientation. It generates tension on the kinetochores, which are being pulled in opposite directions. This tension also signals to the sister kinetochores that it's attached correctly. And each human kinetochore usually binds between 20 and 40 microtubules. For a comparison, a yeast kinetochore only binds one. So now let's take a look at these pictures. What do we have here? We're going to work through these examples. We have pictures of the process of the cell cycle, and we're going to work through each single one so you can understand what's happening. For example, these pictures here are of an onion root tip under a microscope, under bright field microscopy. So this first picture is of a normal resting cell during interphase. So during interphase, remember that the chromosomes are decondensed and the chromatin is undifferentiated. We can see that with the stained nucleus. Remember that interphase includes the S phase, the G1 phase, and the G2 phase, all of which are necessary for the cell to enter into mitosis. So this picture here, this is early prophase. It's called early prophase because chromosome condensation begins to occur in the nucleus, and we can see that in the difference between these two. We see that there's condensing of chromosomes in early prophase, and the nuclear envelope begins to degrade. That's how we can tell the difference between interphase and early prophase. And that will lead into late prophase, where the chromosomes are fully condensed. See the difference between these two cells? It's actually extraordinary, the difference between these two cells. The chromosomes are extremely condensed in this picture. To metaphase. So metaphase, remember, is marked by chromosomes lining up at the metaphase plate. This marks the start of chromosome segregation. From there, we have an early anaphase. We call this early because no cleavage furrow has formed, and we see here that the cells are being pulled towards the poles. And this one would be called late anaphase because a cleavage furrow is forming and a nuclear envelope is reforming around those separated chromosomes, And then we see here we have the cleavage furrow. Cleavage furrow is right there, which is forming at the middle of that cell, which will eventually lead to telophase, where the nuclear envelopes have fully reformed. The cleavage furrow is much more pronounced, and this will lead to the division or cytokinesis, which leads to 2 daughter cells. So that is the cell cycle in a nutshell. We'll be discussing the regulation of it in a future video. Look out for more organic chemistry videos coming soon. I hope you enjoyed this video. If you found it helpful, make sure to like, comment, and subscribe, and we'll see you in the next one. Thank you for watching.